Stanford University.
I'm going to try to get through this in 30 minutes, which might be a little difficult, because I want to have some time to talk about, hear some of your ideas along the lines of the proposal I'm going to make in a few minutes. So I'll explain what I mean first by some of the terms. A Meso-scale computational model is constructed from components corresponding to algorithmic abstractions that explain the activity ensembles of simpler components at the micro, or in our case, the molecular scale. And that when they're combined in different configurations to generate activity on a macro or behavioral scale. So they bridge the gap between the micro, the molecular, and the behavioral. For example, algorithms described in assembly language can explain aggregate behavior at the level of bits and processor opcodes, while also providing a language in which to explain how subroutines are used to manipulate spreadsheets or edit documents. A Meso-scale model bridges that gap. The Meso-scale components are mathematical abstractions that play a role in hidden variables in a machine learning or a statistical model. Ideally, such a model is going to be predictive and diagnostic. Diagnostic is a tall order. Tracing pathological behavior back to its root molecular causes is going to be a tall order. It's surely intractable, and it's most likely just ill-posed. I'm going to provide a model. I've been working for the last 2 or 3 years on exactly this project. And by the way, I started a project 4 years ago in doing connectomics at Google. And that project is doing just fine. That was the structural side of my research in brains. I was motivated initially by the functional side. But the functional side is a lot harder. I think I could ask what I mean by a model of the brain, and I would probably get however many people, 200 different answers from the people here. But I'm going to suggest one possible model. And I have other ideas, and I'll be glad to go into those. But this is the one that's most tightly aligned with Google infrastructure and technology. So scientists have long been interested in the very large and the very small. However, the latter was largely inaccessible to all but the most patient and keen-eyed observers. Albrecht Dürer was one of the few who took the time to explore and render the world of the small accessible to the naked eye. His woodcut prints and watercolors provided extraordinary detail of a hidden world literally right at our feet. The invention and refinement of optical microscopes opened up new worlds. Robert Hooke's micrographia revealed the incredible diversity of tiny animals living in pond water, as well as taking up residence on and inside our bodies. Many of us are familiar with Ramon Kayal's detailed drawings of neurons that continue to inspire and educate neuroscientists to this day. I came upon this drawing in the upper left about 15 years ago, and at the time I thought that it was much more complicated than a real brain could possibly be. I found out just the opposite, that it's not nearly as complicated enough. It was the first rendering I'd ever seen at the time that showed the basic density of the neuropil, the diversity and distribution of synapses, and other features that I had no idea of at that time. The reconstruction in the bottom right is an answer to a question about intracellular actin in the extremities of neurons, and in particular the dendritic spines and smallest processes in the apical dendritic arbors of interneurons. I can bring up the conclusion that I got from Mark Ellisman in a little bit later, if someone's interested in that. Images and 3D reconstructions of this sort will continue to be valuable to scientists, but increasingly we will only be able to look at a small fraction of the data produced by our experiments. We need machines to scan and interpret these large data sets. Inevitably, they will have to be machines that learn, because they will operate in circumstances and at timescales we can't anticipate. Four years ago, we began a collaboration with the Allen Institute for Brain Science to understand the mouse visual cortex. This diagram describes the high-level workflow. First, we employed calcium imaging to record from the genetically engineered mice presented with control stimuli. Then we remove a millimeter cube of cortex, scan it with an electron microscope, and generate an annotated 3D reconstruction, or connectome, of the tissue sample. Finally, we align the structural connectome with the functional calcium imaging data and analyze the resulting data set. My team at Google is working on both the structural and the functional side. The resulting data is measured in petabytes. And when we scale up to the whole mouse brain, which we hope to by the end of this decade, there will be exabytes of data to contend with, unless we can figure out some miracle to avoid even touching the data once. We also share code and analyses with researchers at the Genelia campus of the Howard Hughes Medical Institute and the Max Planck Institute for Neurobiology in Munich. We use machine learning in much of our work at Google, and whenever possible, we avoid bias by letting the data speak for itself. That means that we're a slave to people who produce data. And unfortunately, not like the web, where the data is freely accessible. The data is now in your hands, and we're hoping that you'll share with it, with us and with all your other colleagues, because soon you'll have more data than you know what to do with. The last decade of research in computer vision has taught us that it is more often than not futile trying to invent specialized features to recognize objects or navigate around obstacles. The video you just showed shows an animated tracer that constructs a cell membrane. The tracer is trained to maintain a model of its surroundings and exploit context in deciding whether a voxel is inside or outside of the current neurite. The tracer learns what is relevant and what is not for doing its job. Neural circuit reconstruction is a hard problem. Nonetheless, we expect to achieve higher accuracy than humans and orders of magnitude more throughput. The tracer is controlled by an artificial recurrent neural network. We build and train such networks by combining many specialized components. And these components, while they serve the purpose at Google for recognizing objects in images and videos, will also serve as a basis for the modeling that I'm going to talk about at the end of this, and in the rest of the talk. It'll be at least another year before we have a well-curated and fully annotated connectome for the mouse cortex. In the meantime, we've been working on the common fruit fly, Drosophila melanogaster. We share a fair number of genes with flies, and there are intriguing parallels between human and fly visual systems. The visual lobe of the fly is shown here in red. It's divided into 3 successive neuropils: the medulla, the lobula complex, and the lamina. Several teams are working independently in mapping fly microcircuits. This video is from Jiankang Wang's lab at the Institute of Bioinformatics at Chiao Tung University in Taiwan, and illustrates several trace neurons that span large portions of the fly brain. We need to drill down into the visual lobe in order to see structures of the sort that I've been looking at over the last couple of weeks. The Drosophila compound eye has 800 faceted islets called ommatidia, and the medulla, our focus of attention here, has 800 corresponding 10-layer columns arranged in hexagonal tiles, as shown on the slide. The Genelia 7-column medulla data set is a dense reconstruction of 7 of these columns. Each column has 50 associated neurons. All of them are extensively studied and documented in the literature. In addition to the 350 constructed neurons, there are on the order of 10,000 partially reconstructed neurons that connect to the central 7. The reconstructed neurons comprise a graph, basically the unadorned connectome graph minus annotations. This graph can be compactly represented as an adjacency matrix. And for some mathematicians, that's all they need in order to analyze brains. There are 58 distinct types of neurons represented in the Genelia data set. Neurons can be grouped by cell type to construct a concise account of what types of neurons communicate with one another, as shown in the matrix on the left. There are on the order of 200,000 individual synapses in the 7-column data sets, grouped in 50,000 multi-synapse structures called T-bars. 350 neurons doesn't sound like a lot, especially in an organism with 100,000 neurons. However, this data set is particularly interesting as it includes all of the neurons in this particular piece of tissue. The next few slides will illustrate a typical workflow using some of the tools we've built for exploring such data sets. This slide shows a cloud of points that corresponds to the medullary intrinsic neurons of type 1, the MI1 neurons, and the part of the point cloud shown in green corresponds to the central column. Those shown in, the one in blue is the central column, and the ones in green correspond to the 6 adjoining columns. I fit a line to the central column. The MI1 neurons are particularly interesting because their entire dendritic and axonic arbors are combined in a columnar structure and don't extend beyond a couple of microns from the central axis. Here I've used the central MI1 axis to define a cylindrical volume for closer study. Using this tool, I can selectively turn on specific neurons or classes of neurons, or even neurons that participate in particular subgraphs of the full connectome graph. The 9 panels shown here illustrate tools for analyzing skeletons, examining how pairs of highly connected neurons overlap, and for looking at the distribution of synapses. I'll say a little bit more about the graphs in panels 8 and 9, but first a couple of slides about the graph theoretical and topological analysis that we use in order to analyze these graphs. We're primarily interested in categorizing the microstructure of the tissue sample. The cartoon in panel A depicts the connectome graph embedded in the 3D volume. We run a nonlinear convolution filter over the 3D volume, illustrated here as a 2D tiling in panel B. Despite my inadequate rendering in panel C, the enclosed subgraph for a 3D kernel spanning about 20 microns on a side is generally quite complex. So the subvolume enclosed subgraphs are defined by the position of the synapses and not the cell bodies. It's basically the dual of the connectome graph. Consider the simple network shown in A. The subgraph comprised of cell bodies as nodes, shown in B, is not completely contained in the volume that's bounded by the 2 dashed red lines. Whereas the subgraph shown in panel C, employing the synapses as vertices, is completely contained within the lines. In defining subcircuits contained in local subvolumes, we use the convention illustrated in C. Most of the business is going on in the synapses in any case. There's a long history of work analyzing neural circuits in terms of their graph theoretic properties, though most of the work has been done in fMRI, and in that case, the scale of the voxels or the basic units of resolution are on the order of 5 millimeters, compared with 10 nanometers in the case of the Genelia data sets and electron microscopy data. Intuitively, a network motif is a repeating subgraph that defines a pattern of connectivity that exhibits some degree of functional specificity. We're collaborating with Paul Stolatko, an algebraic topologist at INRIA, on classifying subgraphs in terms of topological invariants packaged as algebraic objects called simplicial complexes. I'm not going to go into the details here, but if you're interested, I suggest you read a great article by David Cox in Archive that provides an overview and discusses some work by Jesty at Princeton. So here's one of those subgraphs. It spans about 20 microns. It doesn't look like it's 20 microns here because in order to make it look more closely to not the dual, but the primary graph, I've used instead the neurons as the nodes, even though all of the synapses are collected in that 20 micron subgraph. This one subgraph involves about 148 neurons with about 10,000 synapses. The simplicial complex consists of all k-simplexes for k greater than 0, where k-simplex is a complete or a fully connected graph, what mathematicians call a clique, with k plus 1 vertices in the unordered graph. And seen as a network, there's a single sink that all of the other nodes drive into. There are actually thousands of simplicial complexes in this small subgraph, and we're talking about 350 neurons total and a very small organism. Understanding these is complicated. If I had more resolution here, you'd see 4 neurons that are typed. By the way, one of the great things about the Genelia data sets is all of the 350 primary neurons are all typed. And there's some typing for the other neurons as well, but that's scattered. So in this small area in the center, I can identify the types of all 5 of the neurons. The one in the 4 of them are drawn as circles, and the one that's drawn as a square is the primary sink. It's an MI1 neuron, which, as we've been told by biologists, is quite common. And diagrams like these we've been able to map them onto the simplicial complexes that exist in these subgraphs. The data that's come and the concepts that have come from the biologists that we've been studying is due to basically decades and decades of painstaking bench work that's now proving extremely valuable for what we're doing. It verifies that the systems and the models that we're building have some basis in what a neuroscientist would consider the fundamental circuits in the gross microcircuit. If the pattern of connectivity in these was more or less random, then we wouldn't find anything that would correspond to a motif. So often what we do is we compare the motifs that we're getting to random graphs to make sure that it's a sanity check. It's not a confirming thing, but to make sure that we're not just picking up noise by doing our analyses. The Genelia data set does not include any connection strength, but one can infer connection strengths by looking at vesicle counts and synaptic cleft measurements and thereby enrich the graph. This is part of what we're doing in constructing the connectomes, is using the same computer vision algorithms in order to make good estimates of these synaptic weights. The K-simplex data corresponds to what are called topological invariants. Maybe if you took graph theory, you've heard of the Euler characteristic. In addition, there's something called the Betti numbers that give you a topological characterization of those graphs. That means in terms of the donut holes, multi-holes like pretzels, and other structures that are basically topological properties. They're invariant to changes in geometry, and that makes them very robust in the same way that the kinds of features that we use for vision are invariant to rotations and other kinds of transformations. We take those topological invariants and we create feature vectors. And those feature vectors are grouped, and then they are clustered using a variety of different things: agglomerative clustering, k-means, almost any clustering algorithm that you apply to those features. And we haven't doctored the features in any way. Basically, we're just giving the clustering algorithms the raw Betti numbers and Euler characteristics and some summary statistics for the simplices that are found within that graph. If you use that in order to come up with a clustering, you get something like this. And these are graphs that are 3D and interactive, and you can rotate them around, and you can basically see the patterns within this in the different colors that correspond to the different parts of the 10-layer matrix, the 10-layer columns, and what's known about their structure and where the action is going on within those columns. The workflow that I've just shown you is pretty common in our lab. And we use it very basically, as I said, sanity checks to make sure that the kinds of things that we're doing make sense. Our objective really is to stand back and let the data talk for itself. In the case of connectomic data, we have a huge amount of data. Not just the petabytes of data that correspond to images of a single brain, but we have fly data, we have worm data, we have data from zebrafish, we have data for birdsong areas of bird cortex. And we have it for multiple organisms. We have literally petabytes of data that we can use at any given time. A mouse brain, the entire mouse brain, is about an exabyte. It's huge. We may not be able to even look at every bit. We may have to figure out some statistical method in order to analyze it. The new microscopes that are being developed by Zeiss and a few other companies, electron microscopes, they have, at this point, over 100 different beams, all of which are slaved to piezoelectric devices, so they're scanning simultaneously. And they say they can gear up to at least 512 in the next year. These are huge fields of view. In addition, scientists like Winfried Denk at the Max Planck, he's developing new kinds of ways of doing the serial sectioning. Imagine a knife that's 2.5 centimeters wide. That's possible today. There's special crystals. They're very hard to come by. You have to grow them individually. These are diamond knives of huge size. That means you could slice the brain of a mouse with one knife. Instead of breaking it up into small 100-micron square or cube pieces, you could do it entire sheet, one at a time. The imaging coming off the back of that and the data coming off the back of that would be on the order of hundreds of terabytes per second. There's no way that we can do anything with that data. We can't even put it on the internet in the sense of just take it to Google server someplace, take it off the back, run a bunch of processes in order to transmit the data over fiber optic cables to Google's data center. literally, just moving that data around would cost tens to hundreds of millions of dollars. Storing it would cost on the same order. So we don't know exactly what we're going to do about that. But the important thing is that we're not going to think about it, that in the sense of we're not going to be doing anything like looking at it, trying to figure out which part is important and what's not important. We're just going to take that data off the back of the microscope and analyze it. So this is a protocol for learning a model of C. elegans swimming behavior. I use the word protocol here because it isn't really an algorithm. It's not something that a machine could perform like a recipe for a cake. Each step requires a trained biologist in order to vet it, to make sure that it makes sense. This is work out of Manuel Zimmer's lab, and Saul Cato was the first author on the paper. The result is a dynamical systems model, and it can be parsed in a simple way. The small box on the lower right corresponds to basically a finite state machine. It's a dynamical systems model, and one of my colleagues at Google, David Cicillo, is one of the world experts in building these models currently working with Shenoy and Newsom and other people here at Stanford. I don't see there's any reason why you couldn't turn this recipe into an algorithm. And in fact, some of my students have done that working in my class last year. I think their algorithms could exploit the connectome for the worm. It happens to be the only full connectome that exists today, although that will change soon because Genelia is getting to the point where they will have a connectome for the fly. I believe that the connectome is essential for learning meso-scale models of large pieces of cortical tissue or tissue of any sort. I don't really believe that the tools of dynamical systems theory are going to scale to larger models, more complicated circuits. And even if they could scale, I'm not sure that they would provide the kind of insight that one would like of a meso-scale model. In some sense, the meso-scale model has to be accessible both to the behavioral scientist, the behavioral cognitive science, and to the molecular biologists who are working at the cell level in neuroscience. At Google, we have lots of models. We have lots of neural networks. Many of them came from work that was done in the 1980s when people got disenchanted with neural networks because they couldn't do backpropagation through multiple layers of perceptrons. When we looked at that about 5 or 6 years ago, we just said, that can't possibly be. It's just a gradient. You just do the math. And so we did the math, and that's how we started building these networks that could scale up to basically billions of parameters and could recognize cats. They recognize a lot of other things too. They're the basis for all of our acoustic models and for the voice recognition systems that we have that work in 60 different languages. They're also not just models that are inscrutable, despite the fact that people say that they're inscrutable and that it is true that we don't understand in great detail models like the inception model, which is about 40 layers, multiple stacks, lots of different loss functions operating at different levels, very complicated. We build those like engineers build tools. We know what each component does. And so we assemble something using some of our intuitions, and then we also run through millions of those models and test them to find the best of them. So here are some of those models. And these models are composed of other pieces. If you think about it, these models came from neuroscience, most of them. Divisive normalization, convolutional networks based upon local field, receptive fields, high-dimensional embedding spaces, distributed coding, zero-shot learning using multiple association areas, recurrent sequence coding layers. All of these either originated from researchers that were knowledgeable about neuroscience, or one way or another, they helped neuroscientists think about structures that they didn't have models for. Sebastian Sung's work in birdsong is a good example of the latter. The other source of power that we have is the combination of both the structural and the functional. The structural basically tells us, it's a schematic, it's a diagram of a circuit. And if you were an alien and someone gave you a processor from one of our computers and you wanted to understand it, and they gave you the schematic too, you wouldn't throw the schematic away. In fact, you might go to some lengths in order to find that schematic. That's what we're doing in structural connectomics. And now if you have pieces or components that are of the sort that I showed you here, each of which individually most of you would understand. You've probably read David Heger's work on divisive normalization. Maybe you've read Sebastian Sung's work on birdsong, in which basically they looked at structures like the basal ganglia, which were able to handle sequences. So you would understand them. And again, you might not understand what they would look like in pieces, but a model that consisted of 4 or 5 of those strung together in some way would be something that would be at least reasonably comprehensible, as opposed to a dynamical system model with bifurcations and fixed points. And maybe the best you could do is a finite state machine that had on the order of 10,000 states. That's not going to scale, and I certainly couldn't understand it, and I don't think that you could either. So what we're going to basically do in this project, or this one of several of the projects that we're working on, is we're going to build systems that look at data that have basically 2 stacks, in some sense, 2 modalities, one structural and one functional. We're going to do convolutional operators that operate on 3D volumes. 3D volumes, 4D volumes, corresponding to the 3D images taken for calcium or voltage indicators. And then our convolution filters are going to be these models. They're going to look for places where they can explain the data that comes from the calcium or the voltage indicators in terms of those models and put them together. In one sense, we do this all the time. When we're doing computer vision, we have all kinds of features. We build up layers of those features, and then we compose them. And our explanation is, well, you know, there's some gradients here that are in this fashion, and there's some gradients here that are in this fashion. Oh, that's a leg, that's an arm, that's a head. They use context. They can determine that, oh, there's a barn there, I can see that. There's a farmer's tractor there. This must be a farmyard. That little black speck down there, that must be the farmer's cat, and the slightly larger one with a red dot in its head, that's probably the rooster. We do that all the time. And I think we can do that in the case of trying to understand the functional structure of large pieces of tech, of brain tissue. It's the kind of thing that Google can do. Each one of these kernels will probably require gigaflops. But if we don't have them now, we'll have them next week. Intel will provide it, always has in the past. And if they don't, we'll just parallelize it in different ways. When we're running our test algorithms, engineers are just fine with starting up a process that runs in 2 data centers and utilizes 2 million cores. And we don't even have to take it away from other things that are running, because basically you're harvesting the cycles that come when an algorithm is busy waiting for data to come in. And for the most part, most of our cores are busy waiting all the time, because input-output is the hardest part and the most expensive part. So that's my story, and that's how we're going to pursue this. And I've gone over my time, haven't I? Oh, okay.
Well, thank you.
Yeah.
Tom, I need you to help me connect some dots.
Yeah.
So you started off with Drosophila and Connectomes. That's kind of an EM level. And but you also had some stuff from C. elegans in there with more kind of block diagrams, and you were talking about connectional motifs, which I think are really, really important. And then we came up to this kind of thing, which, as nearly as I can tell, is kind of a deep learning convolutional net kind of model. And at which of those levels do you think the real secrets are going to lie for us to understanding computational principles of the brain? I mean, I have some biases here, but I'd like to hear your thoughts about that, because you were doing these 3 different levels. It's not clear to me in your own mind how they're connected and what they're important for.
So Saul Cato's worked using dynamical systems theory in order to understand C. elegans, actually a small fraction of C. elegans behavior. Saul Cato's work on C. elegans provides a reasonable model for a small fraction of the behaviors that C. elegans have. But it does a pretty good job of that. And I think it would scale to handle more behaviors. And with better calcium imaging, better tracking, more ability to do optogenetic interventions, we'll do better. And we could probably come up with a reasonable model of that entire organism. But by the time you include all of the other behaviors, that final block diagram I showed you on the lower right, that's going to be pretty complicated. It's going to have all kinds of different types of fixed points, multiple bifurcations. It's going to be very complicated. And so it's going to be like a spaghetti. It's not going to be like something a human being can understand easily. You might be able to use it to simulate something, but that's not what a meso-scale model is all about. A meso-scale model is to provide us with intuitions. It's also required if you want to be able to map things forward and backwards, from molecules to behaviors and behaviors back to molecules, which is a lot harder.
And is this more the meso-scale that you're talking about?
So we need a vocabulary. When you're doing machine learning, you usually use a variety of different kinds of inductive bias. There's no free lunch. You can't do learning without a bias. And so there's a bias in choosing a family of models. And you could use dynamical systems as one family of models. Or you could use something that would be more comprehensible to a neuroscientist. If it happens to be the case that you can explain, say, portions of layer 2-3 in visual cortex of mouse with a half dozen of those structural components that I showed you, that would be a comprehensible model. Because you would understand each of the components, and as long as the wiring diagram between them wasn't too confused, it would be a good model. But now you have to go beyond that. You have to say, okay, now that explains that little piece. What about this other part? And what about what's going on in layer 5, where there's lots of processes coming in from other columns? What explains that? Well, it would probably be more of the same here. It'll be transferring information from one place to another. And a lot of these models, the models shown here, aren't the more complicated things like divisive normalization or max pooling. They're basically just transformations. And you can sort of think of taking one distributed code in one place and making a transformation to another place with matching impedances between the two things by playing with the weights. So that also would be comprehensible. And that's the kind of thing that I'm hoping for. There's another aspect of models, and that is that somehow you need to restrict your attention. And having, say, a library of 10, 20 of these components, that's a restricted model. You can't learn anything either that generalizes beyond what you've seen unless you have some sort of a model that is limited in some way. Doing regularization is one way of doing that. And there are only two ways we know of doing learning. One's called margin maximization. That's what support vector machines do. And the other is called model minimization. And that's the one that we're talking about here. So those are the basic biases and the methods that allow for generalization. David Cicillo uses regularization for that purposes. I think it's better to do it in the model because then you have a little control over the model. As long as your model is still expressive enough, you'll capture the features that you want to, but at least the components will be things that you understand.
I mean, just to amplify a little bit, I agree with you about the dynamical systems. I like the fact that we can start to understand some simple behaviors. I love the C. elegans example, by the way. Also, the stuff going on in the Shenoy lab here at Stanford that David's been part and parcel of at a deep level. I understand that when you start talking about chaining lots of behaviors together in a same system, it's going to get really complicated really fast. Don't know whether we can comprehend it. I like, Dan Yeamans, who's just joined us here, kind of works at this level, and there's promise there. I was really surprised when you went to the EM level with Drosophila at the beginning, because, my own feeling looking at those things is if we can't generalize over some of that detail, then we are hosed. I mean, we are really hosed. But I just want to know, as a computationalist, I mean, you seem to really take that great neuropill slide really deeply seriously, and do you really want that level of detail in the end out of biologists, or not really?
I want to make progress, and I want to make it quickly. My clock's ticking, and I need data. I need data, and I need lots of it. And if I'm right, and you need the structural information that you would get from a connectome, the fly's the place to be for the next 2 or 3 years at least. As we see the calcium imaging scaling up, I mean, right now, calcium imaging only handles, on the order of hundreds of neurons at 30 frames a second. And if you want 10,000 neurons, well, you can get 3 frames a second. And that can be improved, but it will take time. Hopefully less than it has in the past, because due to all the money that's being poured into neuroscience, lots of physicists and engineers and material scientists are getting into this. And so maybe we'll have better tools. Maybe we'll have some Rutherfords out there that will create the tools that we need to make the next big step. But as I see it, the only thing I can get my hands on in the next 2 or 3 years is the fly. I could go back to doing the worm again, but that doesn't interest me. The fly visual system is a complicated system and it has a lot of analogs to humans. And as a result, maybe it will inform the work that I eventually will be able to do with the Allen Institute on the mouse visual cortex. I know, I watch how fast they're moving. I don't think they're going to be ready with, aligned data sets of functional and structural for another 3 or 4 years.
You have one question at the back.
Yeah.
We have one over here.
How you doing? Now, when you talk about creating full connectomes and then transferring them into neural nets, and you touched on a little bit that with the neural nets that we built so far, we understand the structure of the components, but as to all their connections and the weighting between the connections, we don't fully understand the specifics of every single connection. And we found with image recognition in neural nets that over time they build the same filter presets that we find trigger neurons in animals, which leans to say there's a similarity between the structure of how it learned to process the visual data. And would we need, in that sense, if we understand the components enough to build the array to then generate the neural net that will compute the output we desire, would we need an actual full connectome to copy and paste apart for any reason other than preserving the unique neural connection of that connectome? Or would we be able to take a model similar to the computer science side, and once we understand the components, be able to build a system of components that would then compute in the same manner without having to know every connection?
Right. I mean, a Meso-scale model basically is going to create subcircuits. So computers are made out of gates, and then gates are made out of transistors. And the abstractions that we have are half-adders and flip-flops and devices of that sort. I expect we can find those kinds of things using combinations of these, and maybe more sophisticated than that, because obviously these are a lot more powerful. And then you're going to come up with a repertoire of that. The thing is that in order to do that at scale, you have to see a lot of stuff. Not 7 columns isn't going to do it. And replicating 7 columns isn't going to do it either, because you don't have enough data in order to generalize. Another thing you can do with perfectly aligned data, where you know for each of the neurons in your connectome the exact location of the line and the raster in the calcium imaging, then you can basically take the information that you have about the connectome structures, and you can create a time series in which you essentially use the functional data from the calcium imaging at each step in order to create something that will allow you to tile together the combination of structure and function. So I think that's a very powerful model. Henry Markram's done it in some work that just came out recently. But Henry's data comes from simulations, and so it's basically something that's sort of running in the noise. But it has been able to define resting states and other kinds of factors that I guess are symbolically representative of states that people know or understand. Resting state is about the best they can do, and the other thing is, did it crash or not? And if it didn't crash, then maybe it's a little closer to something that a neuroscientist might know. Yeah.
Thank you for a fascinating presentation. If you were to build a sort of process of simulation on this Connectome project, going from the molecular to the behavioral beyond intuitions, and extend that to the nano as well as the neuronal, as well as, say, a Street View level, Google Street View with Time Slider, with Open Simulator, what would be the limitations in the kinds of data that exists now that you would like to overcome in creating this, say, fly brain or similar, that even could extend around us in an augmented reality for various kinds of STEM research?
In the case of the connectomic data, at a microscale, we're seeing circuits billions of times, all with slight variations. Variations in the type of the neurons, variations in the exact way that synapses are formed, variations in slight variations in the wiring diagram, all those kinds of things. The expectation is that a lot of that circuitry is just redundancy in order to deal with hardware that doesn't work very well. Carver Mead said this 3 decades ago. He said that if you want to model something like the brain, then you have to be sanguine about the fact that most of the stuff that's doing computations in the brain doesn't work very well. And the only way that it could work is to combine lots of different things, lots of pieces of circuit that are doing exactly the same thing, that combine in order to make something that's robust enough. So I think that's one of the major hurdles that we have to get over, that there's a lot of structure there that really isn't that important, except for the fact that it's dealing with circuitry that doesn't work very well. And we would never find that unless we had enough data. So it may be the case that we can get to a point so that we can move past that once we sort of understand that kind of statistical model of what the diversity of those circuits would be.
We have one final question.
Yeah, so I have a question regarding the neural networks. So are you using them as, as a potential model that describes how biological neural networks and how the brain works, or as a tool to transform data, a lot of the molecular data, to some other behavioral model?
So these pieces are like the flip-flops or something a little bit more sophisticated. They could be part of a processor that handled multiplication, for example. And well, that's part of, is that on the right direction for you?
I mean, like, are you...
Do you understand what a Meso-scale would be like? Yeah.
I mean, if the neural networks, like, they can both be, like, you can see them as a transformation from data to behaviors. And another approach is to see them as maybe a kind of model on its own.
And they are, yes. One thing is it may not be possible to have a reasonable mesoscale that makes that entire jump across the entire gap from molecules to behavior. My guess is we're going to need multiple layers if it's going to be explanatory in any way. We may need just a layer that takes that statistical view of circuits that are highly redundant just in order to compute in something that's reliable enough over long time scales, over different temperatures of the tissue, over all kinds of other variations. And once they have that sort of robustness, then that's a pattern that you look for everywhere. It's like, that's one transistor. There's 500 neurons there, but it's really just one transistor.
Well, let's thank Tom.
For more, please visit us at stanford.edu.